microbial growth, we typically break the approaches into physical and chemical methods. So an example of a physical method would be heat. Another example of a physical method would be UV, for example. And I just wanted to show you that there are individuals that are using these UV systems, and this particular system here can be dropped in a massive tank and turned on and basically disinfect an entire tank, which you can imagine would be a huge savings in the amount of water that would be necessary to. Obviously, they still would have to flush out residue and from the previous fermentation and things like that, and wash out but you wouldn't need as much hosing down, because most of the disinfection is coming from these large UV units. And so again, if you wanted to look into that further, you can. But UV is great because it's highly damaging to microbial DNA and causes damage to the DNA. It's in the same way that UV causes cancer in people, melanomas and stuff. It does the same DNA damage to microbes. So anyway, UV is becoming has become a pretty important means for managing microbial growth in the industry. But the focus on this slide is for using heat or chemicals, and how we use the concept of a D value to understand how effective either of these things is. And so in this particular case, the first bullet point is a temperature D value. And as you can see, it's both time and temperature dependent for either of these things. And so that's what D value refers to: the time for 90% kill at a given temperature. And so again, imagine, if you had a million bacteria in a flask and you heated them to 90 degrees, and it took 1 second for that 1 million cells to drop to 100,000 cells, then the D value would be 1 second, because it took 1 second for a 90% kill. The chemical D value is the same, but instead of temperature fluctuating, here it's concentration. And so you're looking at concentration of chemicals. And it's maybe easier to see this graphically. So in this particular graph, you see the log number of surviving bacteria on the y-axis and minutes of exposure on the x-axis. And you're determining D value, or decimal reduction time, for a 90% or 1 log reduction. 90% and 1 log are the same in a population. So in this particular case, imagine you have a flask and you start with 1 million cells. So the log of 1 million is 6. This is a log scale. And you heat the flask. You get it up to whatever temperature you want. In my example here, it's 121 degrees Celsius, which is pretty damn hot. But you heat the flask to 121 degrees Celsius, and then you start taking samples from the flask. And so your first sample before heating shows you that you have 1 million cells in that sample. After 1 minute of exposure to that 121 degrees Celsius, you pull a sample out and you plate it on your media, and you find that there's 10 to the 5th bacteria left, or 100,000 bacteria left. You wait another minute and you pull out a sample, and in this hypothetical example, there are now 10,000, and so forth. And so you create this regression. You plot this death rate, and then you can figure out the D value based on just simply eyeballing. In this particular case, any 2 points that represent a 1 log reduction in the population is your D value. So here we have 1,000 cells dropping to 100. How long did it take for that to happen? Well, we extrapolate to our line of best fit, and then we drop down to the x-axis, and we find that the D value is 1 minute. And so you can imagine that this could just as easily be a chemical D value, as opposed to minutes of exposure. These numbers then would just be a certain concentration of drug. Chemical activity obviously is concentration and time dependent. More chemical means more activity, but of course there's a trade-off with human toxicity. You don't want to be making super concentrates and dumping them all over your winery and having your having your workers passing out all over the place. So you have to hit that sweet spot. And of course, time of exposure matters, as I showed you even on that little label for the Lysol spray. If you put something down, you have to make sure it's there long enough before you remove it, so that you can be assured that it's going to actually kill something. So some of the common disinfectants, typically they're oxidizing agents that we use a lot of, things that can damage and cause proteins to clump up. So chlorine-based substances like bleach are usually pretty widely used because they're super cheap, obviously. But one of the downsides is that they're unstable at higher temperatures. So if you want to use a hot water rinse with your bleach, that can obviously cause problems. And the other issue is organic matter can often inactivate chlorine-based disinfectants as well. So it's one of the reasons why jacuzzis, for example, have to be constantly monitored if they're getting high use. So you put a bunch of oily people in a jacuzzi, and it's not long before the chlorine will be inactivated. So, they're not recommended for winery use, these bleach-based cleansers, these chlorine bleach-based cleansers, primarily because of the formation of musty TCA or trichloroanisols. It's possible through the combined action of the mold, corkwood, and chlorine. And so molds can actually grab onto these chlorine-based compounds and convert them into these TCAs. The process can also look something like this, where you have a lignin, which is a common phenolic type compound in wood, in cork tree bark, for example, and then in the presence of chlorine, this phenolic molecule becomes chlorinated, and then the microbes themselves, as you see here, can methylate it and produce this the TCAs. So molds, for example, can methylate it and produce that TCA type compound. But there is something called chlorine dioxide, which I'll talk about, that doesn't appear to contribute to TCA formation, and it happens to be water soluble, so it's a pretty good alternative to regular chlorine bleach. Here's a mechanism of typical chlorine bleach type disinfectant sanitizers. So here's our chlorine, and it's in an aqueous solution and readily forms hypochlorous acid. So that's the HOCl. At an acidic pH, the hypochlorous acid is non-ionized and therefore can penetrate move right through the bacterial or microbial cell membrane. And then inside a higher pH cytoplasm, it can ionize. And so this hypochlorite anion that you see there can start damaging the membrane from the inside out and denaturing proteins from within the cytoplasm. And so this damage allows content of the cell to leak out into the environment, like ATP, for example, or just nutrients in general. It just creates all kinds of havoc. And so here's a little bit of an example of traditional disinfectant working from the inside out, like I showed you. But then chlorine dioxide is actually strong enough to oxidize and damage membranes from the inside out. It doesn't, so it's not as pH sensitive for that reason. And so this graph shows you a little bit of what I just said, where you have percent active biocide on the y-axis, and then as a function of pH on the x-axis. And so you have organic acids. You have inorganic acids like hypochlorous acid and hypobromous acid compared to chlorine dioxide. And so the percent activity of hypochlorous acid, as I just said in the previous slide, drops dramatically as your pH increases. Same with hypobromous acid. However, chlorine dioxide retains 100% activity at a wide range of pHs. And so again, it's a preferable chlorine-based alternative. Another thing that's used, it's not used in wineries. As far as I know, I've never seen anybody use one, but I know a lot of home brewers that use it. They love it because it works really quickly, and they can wash it off pretty fast, and so they can get high levels of killing very quickly. And I think it's used a fair amount in the dairy industry as well, because it can be effective at colder temperatures, and a lot of things are done at colder temperatures in dairy. But anyway, iodophores that contain iodine, but they don't leave residue, and that's important, particularly to home brewers. And they also are really antimicrobial, because they destroy bacterial microbial proteins, and so that leads to rapid death. But they can stain your skin, not surprising, and they react with metals, and so that becomes a problem. There has been I've talked to some people who've said, oh, it's been used as a spray to disinfect bottling lines and so forth. But anyway, they're not probably as important in the wine industry. Ozone's a big one, though. I'm positive that one's used quite frequently, and so one of the good things about it is that storage isn't required. You have basically a device that makes the ozone at the time you need it. But the problem is there's a big upfront cost. I think those machines cost $10,000, $11,000, $12,000 or something, but it seems to be worth it. They're powerful oxidants that can. Ozone's a powerful oxidant that can be more potent than chlorine. Has a really short contact time, so you can spray it on and let it, and basically just let it evaporate, because there's no residue. It just reverts to oxygen, because it's O3, but it reverts to oxygen. It has a great smell too. I love the smell of it. But anyway, it has that fresh, if it's not overwhelming, if it's the right concentration, ozone is probably the reason why the air smells so fresh after a thunderstorm. When the lightning rips through oxygen molecules, it temporarily forms ozone and has a very fresh smell. But anyway, antimicrobial activity is antagonized by organic matter. Kind of like chlorine in that. It is like chlorine in that respect, and it obviously requires corrosion-resistant materials like stainless steel. And it can be used on barrels, which is a good benefit to that. And additional things about its threshold you can read here. The cellular target is typically cell membranes. It's really good at breaking those bonds in cell membranes and causing them to fall apart. It's generally considered safe for bottled, generally recognized as safe for bottled water use, which is great, right? Because then you can justify using it all over your equipment. And it has a very reasonable, effective concentration. And so, something like, it would look something like this.
T1 cards making our beers taste better. Tanks are coming cleaner. There's no potential for contamination. It's an insurance step that I can't.
So you don't need to hear the whole thing, but you can see how it's typically applied. And you saw those guys just spraying it all over barrels and spraying it all over equipment. So in that regard, it's really useful. Quaternary ammonium compounds are also very useful, and particularly because of their longevity. You can spray it on and let it sit for a while, and it remains stable and antimicrobial. So now your applications can be weekly instead of daily, for example. And so that might be useful for spraying it on winery walls or floors where you don't want molds to grow. And so the other thing is the quats are really good at damaging bacterial membranes. And so, again, heat stable, they work over a wide pH range, and they have a very short contact time, which is great. Another big one is peroxyacetic acid. It's a super powerful oxidizer, and destroys lipids and proteins and works at a really low concentration. Another great thing about it is that it has low residual, and the residual that it does produce is basically acetic acid, so a little bit of vinegar, a little bit of organic acid, a little bit of oxygen, a little bit of water, so no big issue there. It's not chlorinated, so you don't get TCAs, and the other cool thing is, unlike trisodium phosphate, it doesn't add salinity to any water, so that you don't have disposal issues. You can dump it without having to worry about it, contaminating the creek or whatever. So another one that's a thing that's used frequently are caustics. Things that like caustics are really good at destroying biofilms. They're highly corrosive, and we also use them to neutralize acidity of spilled wine in a winery floor, because spilled wine in a winery floor over time can be corrosive to the floor or to other surfaces. So it could be used in this way to neutralize. But again, the importance of them is primarily to remove biofilms from surfaces, particularly if I'm talking about like hoses, lines, and things like that. Really important for that. So if you have lines that like beer lines or whatever, where fluid is moving through them. Well, we also use them to clean the labs, but we can use them in lab experiments, for titrations and stuff, if we're doing total acidity and stuff. So caustics can also be used not just to manage microbial growth, but also as reagents in our labs. So one of the downsides of corrosives, those, again, you don't want to use them on certain surfaces. And particularly oak, they're not to be used in oak barrels, because you could really extract a lot of the flavoring agents from the oak and basically deplete the oak's ability to impart flavor. And so that's obviously not a good idea. And caustics, they can be more dangerous to work with. It has a strong potential for side effects. So not surprising, there are caustic alternatives, and so you see this product called Full Clear. After 3 years of using a caustic, you see this pipe all etched and scratched up and damaged. And Full Clear is basically a proprietary product that consists of peroxide and nitric acid, but you can see that there's a bit of a difference there. So in the end, overall, though, the idea is, when you're managing microbial growth, the idea is just make sure you have some kind of validation of your standard operating procedures, using some kind of a sampling technique. Usually it's swabbing or collecting rinse water, for example, for plating or PCR or microscopy. And if you follow those procedures, hopefully you'll have a clean facility and a consistent wine production without a lot of loss from spoilage.